Hello guys, welcome to another video. In this video we're going to learn about the gel electrophoresis. So what is gel electrophoresis? Gel electrophoresis is a method for separation and analysis of macromolecules and their fragments based on their size and charges. It is the gel electrophoresis machine. Here you can see, and there are some components such as electrode, buffer solution, electrode, negative positive electrode, oil, power supply, etc. Then gel electrophoresis is a technique commonly used in laboratories to separate charged molecules like DNA, RNA, proteins according to their size. Charged molecules move through a gel when an electric current is passed across it. An electric current is applied across the gel so that one end of the gel has a positive charge and the other end has a negative charge. The movement of charged molecules is called migration. Molecules migrate towards the opposite charge. A molecule with a negative charge will therefore be pulled towards the positive end. The gel consists of a permeable matrix, a bit like sieve, through which molecules can travel when an electric current is passed across it. Smaller molecules migrate through the gel more quickly and therefore travel further than larger fragments that migrate more slowly and therefore will travel a shorter distance. As a result, the molecules are separated by size. Here are some applications of gel electrophoresis, and the applications are: visualize bands of molecular marker to genotype individual plants, verify amplification by PCR or sequencing reactions, check the quality and quantity of genomic DNA after DNA extraction, separate DNA fragments to clone a specific band. So these are the main applications of gel electrophoresis. Then how is gel electrophoresis is carried out? There are 4 steps. First one is preparing the gel, next one is preparing the DNA for electrophoresis, then third one is separating the fragments, and last one is visualizing the results. So let's go to the first step, preparing the gel. Agarose gels are typically used to visualize fragments of DNA. The concentration of agarose used to make the gel depends on the size of the DNA fragments you are working with it. The higher the agarose concentration, the denser the matrix, and vice versa. Smaller fragments of DNA are separated on higher concentrations of agarose, and while larger molecules require a lower concentration of agarose. To make a gel, agarose powder is mixed with an electrophoresis buffer and heated to a high temperature until all of the agarose powder has melted. The molten gel is then poured into a gel casting tray, and a comb is placed at one end to make wells for the sample to be pipetted into. Once the gel has cooled and solidified, the comb is removed. Many people now use pre-made gels. The gel is then placed into an electrophoresis tank, and electrophoresis buffer is poured into the tank until the surface of the gel is covered. The buffer conducts the electric current. The type of buffer used depends on approximate size of the DNA fragments in the sample. So this is the step of preparing the gel, and the next step is preparing the DNA for electrophoresis. A dye is added to the sample of DNA prior to electrophoresis to increase the viscosity of the sample, which will prevent it from floating out of the wells, and so that the migration of the sample through the gel can be seen. A DNA marker, also known as size standard or DNA ladder, is loaded into the first well of the gel. The fragments in the marker are known length, so can be used to help approximate the size of the fragments in samples. The prepared DNA samples are then pipetted into the remaining wells of the gel. When this is done, the lid is placed on the electrophoresis tank, making sure that the orientation of the gel and positive and negative electrodes is correct. We want the DNA to migrate across the gel to positive end. So this is the step of preparing DNA for electrophoresis. Then next step is separating the fragments. The electrical current is then turned on so that the negatively charged DNA moves through the gel towards the positive side of the gel. Shorter lengths of DNA move faster than longer lengths, so move further in the time the current is run. The distance the DNA has migrated in the gel can be judged visually by monitoring the migration of the loading buffer die. The electrical current is left on long enough to ensure that the DNA fragments move far enough across the gel to separate them, but not so long that they run off end of the gel. So this is the separating the fragments. Then last step is visualizing the results. Once the DNA has migrated far enough across the gel, the electrical current is switched off and the gel is removed from the electrophoresis tank. To visualize the DNA, the gel is stained with a fluorescent dye that binds to the DNA and is placed on an ultraviolet transilluminator, which will show up the stained DNA as bright bands. Alternatively, the dye can be mixed with the gel before it is poured. If the gel has run correctly, the banding pattern of DNA marker standard will be visible. It is then possible to judge the size of the DNA in your sample by imagining a horizontal line running across from the bands of the DNA marker. You can then estimate the size of the DNA in the sample by matching them against the closest band in the marker. Here we can see the visualizing the results of the sample. So this is all about the gel electrophoresis. That's all for today. See you in the next video. Thanks for watching.